Oyster Point Pharma is a venture-backed startup pharmaceutical company located in Princeton, New Jersey. Our focus is to address the loss of tear film homeostasis in dry eye disease. And we do that by stimulating the trigeminal parasympathetic pathway. The trigeminal parasympathetic pathway has been known to be important for basal tear film production as air passes through our nasal passages. And we know that this pathway is also responsible for innervating the lacrimal functional unit, which includes the meibomian glands, lacrimal glands, and goblet cells. We have 2 molecules in phase 2 clinical development. I'm going to show you clinical data from 3 phase 2 studies today, 2 of which will be presented here for the first time. These molecules are nicotinic acetylcholine receptor agonists. They're delivered as a simple aqueous and preservative-free nasal spray, and they target the anterior portion of the nasal cavity, in specific, the inferior turbinates. Here's a video showing the patient using the product. You'll see that a nasal spray is administered into each nostril. The drug then binds to the nicotinic acetylcholine receptors on the trigeminal nerve, thereby causing depolarization of the nerve by calcium ion influx and stimulation that's sent to the central nervous system. The lacrimal functional unit is then stimulated by this pathway, and so we get production of lacrimal gland fluid, we get production of meibum, and production of mucin from the goblet cells. Here are patients being treated with the product. You'll see OCO1, our first product on the left side, and OCO2 on the right side. And within 10 to 15 seconds after delivery of the nasal spray, you'll see that tear film height build up in the lower lid margin. So the drug acts very quickly, provides good tear film. This is the first study that I'll be presenting today. This is the first time we'll present the onset study. This is a large randomized masked controlled study, a dose ranging study delivering a high, medium, and low dose of our first product, OCO1. You'll see that our pre-specified primary endpoint was the mean change in Schirmer score at week 4. We had 2 pre-specified secondary endpoints, both eye dryness score at week 3 inside the controlled adverse environment chamber and eye dryness at week 4 outside of the chamber. Here are the primary endpoint data. You'll see that we achieved statistically significant improvement in Schirmer score at all visits and with all doses. Inside the controlled adverse environment chamber, there was also a statistically significant reduction in eye dryness score at 5, 10, and 15 minutes, showing how acute this product works at providing systematic reduction. And here you see the eye dryness score data at week 4, and you'll see that we had statistically significant improvements in the 0.1% dose group. Here are the safety adverse events. You'll see that the most common adverse event was sneeze after any instillation. These were all mild in severity, transient and self-limiting, and usually within 30 to 60 seconds after delivery had subsided. The second study is the PEARL study. This is a previously presented study, also a dose-ranging design. You'll see this is now with our OCO2 product, so our second clinical asset. We treated with a high, medium, and low dose drug here as well. Our sign endpoint was Schirmer score. Our symptom endpoint was eye dryness score in the controlled adverse environment chamber. Again, you see that we achieved statistical significance at all 3 dose groups with a very nice dose response. Similarly, when we look at reduction in symptom score, we see a statistically significant reduction in symptoms at the high and medium low dose groups. The last study that I'll be presenting today is the Rainier study. This study is now also with the OCO2 molecule. You'll see that this is a smaller study. Patients were randomized 2 to 1 with treatment group versus placebo. Our primary sign endpoint was mean change in Schirmer score. Although the study was not powered to show statistically significant changes in symptoms, I will show you that data today as well. Here are the results. You'll see that we achieved a statistically significant improvement in Schirmer score at week 1, week 2, and at week 4, that value was not statistically significant, although you will see that the drug responded favorably compared to all other visits. There was a directional improvement in symptoms, but like I said, because of the small sample size of the study was not powered to show statistical significance. This was seen not only outside of the chamber, but as we see in the Pearl and onset studies, also within the chamber. Adverse event profile is very similar to what we see in the onset study. Again, all mild in severity, transient, and self-limiting. One of the important things to point out here is with this product, it's ocular surface sparing, so we do not see ocular adverse events, nor do we see systemic adverse events related to this compound. So in summary, we're developing 2 molecules to treat the trigeminal parasympathetic pathway to promote the patient's own tear film. We look forward to moving to phase 3 in the middle of 2019. Thank you.